So up until now, we've been laying the foundations for microbiology. We've learned about basic biochemistry, we learned about structures of the cells, and we've even learned about some of the reactions that go on inside of the cell. Now that we know these things, we can apply them to the rest of our semester. Things like, how do microbes grow? How do we stop them from growing? Ultimately, we're going to do things like learn about disease and even the immune response to microbes, based on the many things that we have just talked about. Module 5 is all about microbial growth. We're going to talk about some of the basic factors that are required for microbes to grow, ultimately such that if we know what they need to grow, then we can prevent them from growing. Okay? However, warning, math alert. You know, in biology, we don't like to do too much math, right? But math is a necessity sometimes. Sometimes numbers tell the story. So I can already feel the anxiety rising just a little bit. Blood pressure is going up just a little bit, right? A little bit of a panic attack coming on, but that's okay. We'll get through this. We're going to take it nice and slow. However, it's going to spring on you at any moment. 1 second, you're going to be writing notes. Next thing you know, numbers appear. So let's put that aside for now. Try to focus on the content at hand, and let's talk about microbial growth. Now, typically when we talk about growing, we're talking about getting bigger. You see your kids growing and say, oh my goodness, you're so big, you've been growing. What have you been feeding them? All right, but for microbes, it's not quite the same, because microbes in general, they don't really increase that much in size. Of course, older cells will be a little bit bigger right before they divide, and after they divide, they're a little bit smaller. But in general, we don't talk about microbes growing in the sense of getting bigger. We're talking about increases in cell number. Let's do it this way. Increase in cell number. There we go. Increase in cell number, right? So that's generally what we talk about when we talk about microbial growth, right? So growing microbes then really means an increase in population size. So, we want to learn how to talk about microbes with respect to populations as opposed to size of cells, right? So you can actually have instances where cells are dividing, but they're not growing because the population number is not increasing. Now, of course, it's important to understand the conditions that are necessary for microbial growth, because if we know what they grow or how they grow, the conditions required for growth, we can limit their growth, right? So then we can use it to prevent things like food spoilage, disease, et cetera, et cetera, et cetera. And of course, sometimes we want to encourage the growth of beneficial microbes. Sometimes we want certain microbes to grow, right? So then again, understanding their conditions, we can then use things or do things such that they can grow favorably amongst all the microbes that are present. Sorry. When we're prokaryotic reproduction is always asexual, right? So they don't really do any sexual reproduction, which means then that all daughter cells are genetically the same, and that's how we refer to prokaryotic cells, right? The cell before it divides is called the parental cell or the parent cell. And then after dividing, we have two daughter cells, right? And because reproduction is asexual, all daughter cells are genetically the same. There are mechanisms for genetic recombination, right? So microbes are able to vary their gene combinations to some extent, right? But we're not going to talk about today. We're going to save that for Module 7. Cell division requires DNA replication first, of course, right? So you can't just divide cells. So along the way, DNA needs to be copied. Very important anabolic process, right? But again, we'll save that for a little bit later. But just understand that it's not as simple as just dividing the cells. Even though we say prokaryotic reproduction is very simple, it does require some things. In terms of actually dividing the cell, binary fission is what bacteria use to divide. So it's called just binary fission. And at its simplest, really, we have two cells. This one here is just about to divide. We have this thing called a cleavage furrow, where if you almost imagine like a finger and a thumb pinching these two in half until this thing kind of comes together, a new septum or a wall forms, and then we have our two daughter cells. There's a very important protein involved in this, FtsZ or FtsZ. That's very important for dividing the cell. It forms a Z ring that really kind of brings the two cells together, so rather than a pinching, now think of it more as a pulling, right? We have attachment on each inside of the middle of the cell, and then the FtsZ ring starts to pull the things together. A wall forms. We got to build new membrane. We got to build new peptidoglycan. And then when that thing comes together, now we have our two daughter cells. So the thing about reproduction is that we can measure this, right? So there's something, and we define this term as generation time because all things being equal, the time it takes for a cell to divide is going to be the same, right? So we call this generation time. Sometimes we call it doubling time, right? And again, it's just time it takes for once the two daughter cells are formed, how long does it take for that one of those two daughter cells to become two daughter cells, right? So if we want to think about this again on a population scale, really what we're talking about is the time it takes for a microbial population to divide, aka double, right? So really that's all that happens. Within whatever the generation time is, the population is going to go from one cell to two cells, from two cells to four cells, etc., etc., etc. It really does vary among species, so there's no such thing as this universal doubling time or generation time. We often say bacteria divide really fast, but not all of them do. Some of them divide really slow, right? But under ideal conditions, E. coli growing in the lab under a very rich food source with plenty of oxygen and plenty of oxygen, plenty of nutrients. Doubling time can be as short as 15 to 20 minutes, right? And you take something like E. coli under those conditions, and then in this case, populations, they do grow incredibly fast. One single E. coli cell will become 1,000,000 in 20 generations. That's 1 to 2, 2 to 4, 4 to 8, etc., etc., etc. Do that 20 times, and then you're going to have just over 1,000,000, right? And that takes about 6.7 hours with a 20-minute generation time. After 30 generations, you're over 1 billion. Right, and that's just over 10 hours, right? So under ideal conditions, you can have a single E. coli cell become 1 billion in less than 10 hours. We are going to go talk about some math, right? So if your math is not great, and that's okay if it isn't, it happens, right? Appendix B has some math concepts that you can cover, right? So the basic ones here, I think that are most relevant are multiplying and dividing by 10, scientific notation, and then logarithms, right? And these are just enough to understand some of the things we're going to talk about. So I'm going to speak like you have reviewed this, okay? If you have not reviewed this, and then some of the things that I'm saying don't quite make sense to you, review, go to Appendix B in your book, all right? And then you can come back and revisit the lecture. Okay, so as long as conditions don't change, growth is going to be constant, right? So, again, 1 becomes 2, 2 becomes 4. This actually means that you can predict how many cells there are going to be at a certain amount of time. Or if you know how many cells there are now, and you know the doubling time, then you can figure out how many cells there were, right? And then, so this kind of works both ways. There is a mathematical formula that you can use. It's n subscript n equals n subscript 0, 2 to the power of n, where n subscript n equals the number of cells there are now, n subscript 0 equals, or n naught, equals the number of cells that we started with, and then little n simply refers to the number of generations, right? The number of times a cell has divided. However, we're not going to use this formula, right? So right now, and we can look at it. You should recognize it. And there are variations of this where sometimes we'll factor in things like t or time. But as far as we're concerned, let's ignore it, okay? Because we're going to keep our math super duper simple, right? Based on this concept of doubling time. Now, growth is what we always say exponential or logarithmic, right? Because it goes real fast, right? So sometimes we don't understand what a logarithmic or exponential growth is. So basically what we're doing is we're going to double every single time, right? So we're multiplying our population by 2, right? So that's what exponential means. We're multiplying things as opposed to a linear growth. Linear growth is simply adding, right? So 1 becomes 2, 2 becomes 3, 3 becomes 4. That will be linear. Whereas exponential will be 1 becomes 2, 2 becomes 4, 4 becomes 8, and we're doubling every time. Okay, I warned you. Here comes the math, right? So this is a very standard type of question. We have a defined doubling time of 30 minutes. starting population of 1 times 10 to the 5 cells. How many cells will be present after 2 hours? Okay, so again, you could use a formula if you wanted to. You can go dig it up somewhere, right? But sometimes formulas are complicated and you have to use exponents. And, if you don't know how to use a calculator very well and you're punching in things funny, you get these weird numbers. And the problem with that also is you're kind of trusting that you punched in the numbers right. You're trusting the formula to produce a number. Whereas if you work it out, it becomes very simple. Okay, so let's do it this way. This is how I like to start these type of problems. I like to write down my my knowns, right? So we have a doubling time, a G of 30 minutes. Oh my goodness, this is supposed to work better. And then we have our starting population, right? That's our N0 or N0 equals 1 times 10 to the 5, right? And then we have time, oops, that's supposed to be time, of 2 hours. Okay, so let's do this then, right? So I like to write down, oh yeah, and then we're looking for final N. So N at the end equals what? Okay, so I like to start now with the beginning. So here is what we have, 1 times 10 to the 5, right? Just like that. And if you have trouble with that, that's simply 1 with 5 zeros. So we can just go like this, 1, 2, 3, 4, and 5, right? However you want to do it. 1, 2, 3, 4, 5. Okay, good. I thought I punched that in wrong. And then now we're going to pretty much just double it, right? Because remember, what happens after 30 minutes? Well, after 30 minutes, we should have doubled cells, right? So let's do that. I like to draw process. We have an arrow. That's supposed to be an arrow. We have 30 minutes. And then what's going to happen after 30 minutes? Well, we're going to have double that, right? So 200,000. That makes sense, right? Because after 30 minutes, we're going to double. Remember, we're going to do this for 2 hours, so we need to double again. So what happens in the next 30 minutes? So now we have 1 hour, right? In the next 30 minutes, we're going to have 400,000. Makes sense, right? Because we doubled. And you don't have to do this in your head. If you want to, you can go 1 times 10 to the 5 times 2 in your calculator. You can go 100,000 times 2 in your calculator, whatever you want. It doesn't really matter. No shame, right? And then we are still not at 2 hours yet. So now we're going to go 1.5 hours, right? And then this will now be, again, I'm putting in my calculator, 400,000 times 2. Now we're going to be at 800,000. And then another 30 minutes, oh, look at that. We're now at 2 hours. So this will be our final number. We are now at 1,600,000. And if you did it the scientific notation way, that would be 1 times 10 to the 6 cells. And so that will be your final answer, right? Couple of things to point out here. Couple of things to note. Sometimes we count, because, in 2 hours, we know we're going to double this thing 4 times, right? So 2 hours is 120 minutes divided by 30. That's going to be 4 times. So we know there's going to be 4 doublings in 2 hours, right? Sometimes what people do is they'll count the numbers here. So they go 1, 2, 3, and 4. And then they'll end here at this number, right? But that's not the right number, right? Because remember, it's doublings that we have to count. So I like to count the arrows. 1 arrow, 2 arrow, that's 2 doublings, 3 doublings, 4 doublings, final answer right there. Okay. And then just multiply by 2. Take your calculator, punch in the number, right? Times 2, times 2, times 2. You keep on going until this number above your arrow is this number right there. Okay. So using this basic formula, we can now figure out many things, right? So for example, let's say we had this exact same number, right? It's the same situation, right? But slightly different problem, right? So let's say the question read, you have a starting population of 100,000, or 1 times 10 to the 5. Final population of 1.6 million, or 1.6 times 10 to the 6. Right? Took 2 hours to get there. What's generation time? Well, what I would do is I would start with my initial population. I would just keep doubling. Bang, 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 until I get to my final number. Right? So I would have just kept doubling until I got to 1.6. And then I count the arrows. 1, 2, 3, 4 arrows. Right? And then you take your 2 hours, divide that by 4, the number of arrows. So then if we have 4, because remember, each arrow represents a generation, right? If we have 2 hours per 4 generations, we then end up with 30 minutes per 1 generation. All right? Similarly, let's say you had a final population of 1.6 that's been grown for 2 hours, generation time of 30 minutes. What was the initial concentration? In this case, then you would, once again, do this, and then this time divide by 2. Divide by 2, divide by 2, divide by 2, divide by 2 until you get to your 2 hours. And you keep dividing, and then you'll end up with your initial concentration. All right, so we have 4 numbers here. You can just keep playing around with various things to try to get to where you're at. Okay, that wasn't too bad, right? So we're trying to keep the math simple, right? Again, don't use formulas. Formulas can be complicated. Right? We're just going to keep things nice and simple. Main concept here is doubling time. Each generation, population will double. Okay, so that's kind of what we want to focus on here. So bacteria are going to grow, right? And because bacteria grow so fast, right, we plot these things on what we call a semi-logarithmic graph, where one scale is linear, one scale is logarithmic, right, which means it goes up real fast in numbers. And the reason for that is on a linear graph, the numbers just increase so fast that everything's going to be compressed, right? So, and we'll see the 2 graphs side by side, and then see how the log scale is so much more efficient for measuring growth. Just a little bit about the log scale, if you don't quite understand it. It increases in increments of 10, right? So each major line is basically a 0, right? So one major line will be 10, next one will be 100, and 1,000, et cetera, et cetera, et cetera, right? And whenever you're doing anything that's growing logarithmic, definitely when you're doing populations, you want to use a log scale, just because of the fact that, again, we're going to see the effect that having a log scale has, right? And then our x-axis is going to be linear, because as far as we're concerned, time is linear, right? I think time is still linear. So here we have the exact same data points, one on a linear scale, one on a semi-log, right? So here's our linear time on the bottom, right? So notice that's the same. And then here we have our log scale. And actually the scale is the same, right? We're going to go up to, that's 1,000, that's 1,000, right? If you don't know how to read this, by the way, 10 to the power of 3 is simply 1 with 3 zeros. 1,000, right? So then this here would make it, what number is this? Right, a 1 with 2 zeros, 100. All right, and of course here is 0. Whereas here, notice the scale difference already, right? So this teeny tiny little gap here is 100, right? Over here on our log scale, 10 to the power of 1, right? That's 1 with how many zeros? 1, right? So that's actually 10. Here's our 100. And then here's our 1,000. Right? So notice how now our distance between, by the way, this is 1. It's actually not. So a 0 is actually 1. So there's no such thing as a 0 on a log scale. I know, weird, right? So we're going to just ignore that if you don't understand that concept, right? But this jump of 10, right, is the same from 1 to 10 is the same as the jump from 10 to 100, which is the same from 10 to 1,000, right? So it goes up in these equal jumps of 10, right? Whereas here we have a linear thing, right? So in this case, we'd have like 100 little lines to get to 100, and then here we have 1,000 little lines to get up to 1,000, okay? Because of the fact that growth starts slow and then gets rapid, linear scales actually make it really hard to see increases in number, right? So here, again, we have the exact same data points, right? So this, after 1 hour, we have a population of 2 cells, right? So right there. So we have 2. And remember, that's like 100 lines here. So 2 would be pretty much right about 0, all right? So as far as we're concerned, actually, from here to here, the population, it hasn't grown, because we haven't seen an increase, right? After 5 hours, right? So then 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32. So after 5 hours, we now have a population size of 32. And again, on this scale, 32 is going to be right there, right? And again, it's really hard to see this increase, right? And then after another 5 hours, we now have 1,024. All of a sudden, now we have this straight line. So if you're looking at this visually, it looks like growth kind of happens during this segment right there. Right? So if we're looking at this saying, oh, it looks like the population started growing after maybe 3 or 4 hours, because that's where I see the increase. The other issue we have with this is that how fast is it growing? It's really hard to figure out some sort of rate because of the fact that it's this curved line, right? Humans, we don't like curved lines. We like straight things. Much more easier to see. Let's look at our data point over here, right? Remember, this is 1, so that actually is 2, right? So after 1 hour, we have 2 cells, which is way up here already. So actually we can already see after 1 hour, we can see an increase from 1 to 2, right? So that's visual, right? So we can actually say even definitively after 1 hour, it's like, oh, look at that. We have an increase, we have growth. And then this is where things get really powerful. Look at that. At the 5 hour mark, our 32 cells, look how high that is, right? So we can actually see that as well. Oops, I maybe should do this. I don't know if you're actually seeing that pointer. But anyways, look how high that is. We can see that difference, right? Because that's immediate growth. And then at 1,024, we see that as well, right? And notice this line is this nice linear straight line, right? So when we're looking at this, we can do a few things. First of all, we can tell immediately that we are seeing growth, right? And we can see that growth has started right from the beginning. We can see that growth is continuing there, and then it's actually continuing there. We can see that growth is constant. The line is this nice straight line that doesn't waver. And then the other beauty of this is that we can see the rate of growth. So let's say we have this population and then a second population. That's supposed to be a straight line. Let's pretend that's a straight line. We have a second population over there. Like, even without me saying anything, you can already see that this population is growing faster than this population because we can see that slope, right? It's very powerful. Right, so again, just kind of zooming in at this linear scales, it makes it really difficult to see small increases. Definitely, if we're looking at another curve line, let's say we have a population that started there and we see something like that, right? Which one is growing faster? Which one's growing the same? I don't know. It's really hard to tell, right? There's some random line that I drew and it looks the same, but it's really hard to tell, right? Whereas on a semi-log graph, any increase in number is seen as a straight line, right? So now let's say I have another second population that starts here, right? And I do that. That's funny. So here you can actually tell, even though the lines look very similar, right? Look at the slope. It's actually a little bit bigger there, a little bit smaller there, right? So I would say actually that this one is growing slower than that population. Right? And you can see that because of the slope of the line. So that's how we're going to plot these things. A little bit challenging to kind of plot, right? Using the scale. Most important thing is just understand that, there's going to be 10 lines here. And then each one of these is a jump of 10. So remember, what is this again? That's 10. This one here, that's 100. This one there, that's 1,000, right? And then we're just going to go up with those lines, right? So starting with, remember, this is not 0, it's 1, right? So this is actually the reason that's a 0. Fine, I'll just go there. It's that this is 10 to the 0, and then if you know anything to the power of 0 is just equal to 1, right? So that's 1, and then we just count 2, 3, 4, 5, 6, 7, 8, 9, 10. Right? Now we have 10. Now we're gonna go in tens. 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. Right? 100, 200, 300, 400, 500, 600, 700, 800, 900, 1,000. What's the next line here? 2,000, 3,000, 4,000, et cetera, until we get to 10,000, right? So that's how one reads one of these semi-log scales, or sorry, this log scale. Okay, so we now know how to plot microbial number, right? So it turns out that when you grow bacteria in what we call a batch culture, right? A closed culture where you kind of start with the food that you start with. The waste that's produced is the waste that's produced. you're going to have this characteristic growth pattern, often called the bacterial growth curve, because we almost always do this with bacteria, right? But it doesn't have to be. We can do this with any single-celled organism, right? Almost any organism. I'm just going to say that, right? Why not? But, we almost always do this with bacteria, right? So it's often, again, referred to as the bacterial growth curve or just the growth curve. And when we do this, we have 4 distinct defined phases. The lag phase, log phase, sometimes called exponential, stationary, and then death, or sometimes called decline. This lag phase is defined by this period of no growth. A log phase is a period of rapid growth, right? Log as in, like the log graph. So you're going to see this rapid growth, rapid increase. Stationary phase, now our growth has stopped. Remember, that means population is not increasing anymore. And finally, death is a period of decline, right? So let's draw this out before we even look at our graph, right? Although some of you may have already seen the picture, but that's okay. That's supposed to be a graph, right? So on a graph, based on what we just know, what would a period of no growth look like? Right, straight line, because we should see that, right, as a straight line, right? So good. The lag phase then should look like a straight line. If now we see a period of rapid growth or an increase in number, what will our log graph show us? It should show us an increasing straight line, right? Once again, now we're hitting a period where growth stops, so we should see no growth. Once again, then that should be a straight line. And then our death period of population decline, right, which then will be the opposite of growth, which then means that things are going down, right? Look what we just drew. Look what our growth curve looks like. Just what we drew, right? A couple of things about each one of these phases, right? So we kind of define these things as kind of what's happening, but it's a lot more active, a lot more busy than the numbers suggest, right? So in lag phase, even though there's no increase of in number of the living cells, the cells are metabolically very active, right? So you think of this as this acclimation phase, this period where the cells are kicking around and they're like, okay, how warm is it in here? What's the temperature like? What kind of food have they served us? Do I need to make special enzymes to eat this food, or can I just eat it with the enzymes I have right now? How many of us are here right now? Are we going to be crowded right now, or is there a lot of space? Do I need to speak with anyone to grow? All right, so there's a lot of things going on. So, these cells are constantly kicking out these enzymes, very high in anabolism, and just getting ready for growth. What they're not doing is dividing, all right? So, and that's why we see this straight line. So there's no cell division going on, all right? But metabolically, very active. Once they're ready, bang, they're going to divide, right? So in the log phase, they're going to actively divide. All cells are pretty much metabolically similar. They are all kind of synced up, right? Maybe right at the beginning of log, they might be slightly different, right? But once they're churning out, they're going to start dividing like crazy, right? And then their focus here is going to be on catabolism. They're going to just burn all the sugars, all the food sources that they have present. And then anabolism, they're going to divide as mad as they can. And then here is where we calculate G, right? So G, or the generation time, is going to be defined during the log phase, okay? In stationary phase, now as the population is starting to increase, right? Remember, when you eat, you have to poop, all right? So as the cells are starting to grow, a couple of things is happening. First of all, food is going to start to disappear, right? So there's going to be less food around. So the cells are going to have to now start, they're not getting quite as much nutrition as they once got. The other thing that is happening is that waste is starting to build up, right? And waste is going to be toxic, right? Imagine you living in your room for 30, 40 generations, right? And you got to poop in there somewhere, right? At some point, that poop is going to start building up to this level where it's going to start affecting you, right? So in stationary phase, now what's happening is that cells are starting to die. Cells are still dividing, right? They're still going to continue to grow, but the rate of cell division equals the rate of cell death, approximately, right? So for every one cell that dies, one cell is going to divide. So that's why we see this plateau in number. So cells are still metabolically active. Cells are still dividing. However, population has stopped growing, right? No increase in number. During this time, cells are now, because they're stressed, they're starting to do some interesting things. So many times here now, they're going to do things that help their own survival. So they'll often make these things what we call secondary metabolites, things that maybe help them get like squeeze a little bit of energy out of some extra type of things that might be around. Sometimes they'll start to put out some chemicals that'll kill off their competitors, right? They'll do these weird things during this phase. Sometimes they'll even start kicking out DNA and try to see if they can like, hey, maybe this DNA is slowing me down a little bit. Maybe I can then try to kick it out and see if I can speed up my reproduction just a little bit, right? So there's a lot of interesting things that go on in stationary phase, but remember, cells not really dividing, right? And then finally, the death phase. At some point now, the food supply is so low or the waste is so high that cells are now starting to die faster than they are dividing. Right? And so we start to see this decline. And at some point, either the population will completely die out, or there are a small number of what we call persister cells that can really slow their metabolism down so much that they can remain dormant. And I just read this interesting article somewhere in the South Pacific. I don't know where because I didn't read that deep. They took some sediment from about 75 meters below in the ocean near these hydrothermal vents, and they just scooped up some sediment that was apparently metabolically inactive, right? But using the right kinds of foods, they were actually able to coax out life from bacteria that had been dormant for, wait for it, 1.2 million years. Can you believe that? These things were dormant for 1.2 million years. And then they were able to kick them back, right? Going from this death phase or this dormant phase, eventually right back into their log phase, right? Isn't that crazy stuff? So they will sit there for as long as they need to. And that's like the wonder of bacteria. So many interesting things that bacteria can do, right? Okay, so lag log, stationary death, right? Your very characteristic bacterial growth curve. When we're talking about every now and then, right, the bacterial growth curve is not good for us, right? We want to have this continual growth, right? So typically in industrial applications, when you're using microbes to make food for you, to make vaccines, to make therapeutics, because many of these things can be engineered to do that. So then in order for these things, if you want to have these microbes growing continuously, you need to do one of two things. You need to do not one of two things. You need to do two things. You need to supply this constant supply of nutrients, right? So feed goes in, and then you need to be able to get rid of waste, right? Or the effluent, right? So, and then in this chemostat, what you have is this aeration, right? So this big mixer in the middle here that provides oxygen, right? And then you're going to, at some slow rate, you're going to put this fresh nutrients in, and then you're going to suck stuff out, right? And then really the key here in engineering is to regulate the feed in and the effluent out such that we are constantly staying in log phase. Because remember, log phase is what we want to be in. We don't want to get to stationary where they start doing weird things. We definitely don't want to get to death phase, right? And of course, in lag phase, they're not even growing. So we want to maintain them in log phase. This thing here is called a fermenter, right? An industrial fermenter. Now, keep in mind that's different from our catabolic fermentation, right? So, in fermentation, we're talking about this growth where they use this organic final electron acceptor, produce some sort of organic waste product, right? We just learned that in our last module, right? But from an industrial application, a fermenter is this vat where we allow this continual microbial growth. Okay, if you want to take a break, now will be a good time to take a break, right? Because we're going to go and start doing some stuff that may or may not involve some numbers, right? So take a break now. If you want to if you already copped out and took a break a little bit earlier, well, I guess you can just keep on going. And if you just want to power through, let's do this, right? But you should get some paper and pencil or pen, whatever you want. Actually, I think I left you space on the note papers to be able to follow along, right? Because we're going to do some scribbling. Okay, so we have this bacterial growth curve. One of the things that's kind of implied is that we can count the amount of bacteria, right? What we call the bacterial load, right? How much bacteria is there within some various sample, right? And we can have it in our growth media when we're growing them in the lab. But we also need it in things like, when we're measuring water quality, for food safety, environmental surveys. Sometimes we need to know how much bacteria there is, right? So we need to figure out then a way to do that. And each of these applications is going to require certain type of counting method. Two major approaches: the direct method that involves counting cells of some sort, counting growth of some sort, right? So we are able to visualize the growth, right? Whereas an indirect method is an estimate based on some sort of non-counting method. Right? Well, what does that mean? Well, that's okay. We'll get there, right? And, obviously the direct method sounds like it's the best, but sometimes it's not the most useful, right? And there are advantages and disadvantages to each of these methods. Okay, and we'll talk about these, both of these, and we'll talk about specific direct and indirect methods. Okay, do we have our scratch paper ready? I hope so, because it's gonna get real pretty soon. So we're gonna talk about these three direct methods: direct microscopic cell count, a viable plate count, and then most probable number. And just looking at the names, we can already see a couple of things here. Look at that, count. So we're gonna be counting some things here, right? Directly counting cells, and in here we have some sort of number, right? So it'll be some sort of quantity or quantitation of things. Okay. We'll start with the direct microscopic cell count, or sometimes just called a direct cell count. This is probably the simplest way, really. I mean, if you were to say, hey, why don't you figure out how much bacteria there is? Like, what's really the simplest way to do this? Well, you're just going to take something, stick it under the microscope, and then count it. All right? And that is pretty much exactly what this is. You're going to take your sample, you're going to put it in the microscope, and then you're going to count it. Now, for counts to be useful, you need to know how much sample you have. You need to know, of course, how much bacteria there is. And then you need to be able to figure out then what we call this concentration, right? Because you need to know this quantity of microbes based on the sample size, right? So in order to do that, we have something called a Petrov-Hauser chamber, right? It's this calibrated slide that's used to count. It has a number of different grids, and each one of these are slightly different, right? But they always tell you what the measurements are. So in this case, this big square, and they're all squares, by the way, right? This big square here is 1 millimeter by 1 millimeter. Oh my goodness, not only are we doing math, we're going to use metric, right? Don't worry about it. Ignore it, okay? So 1 millimeter, 1 millimeter, 1 millimeter. If you use these squares here, these guys are 0.25 millimeters by 0.25 millimeters. And then if you're looking at this grid over here, these tiny little ones here are 0.05 millimeters, right? So we're going to blow up this one section there, which means then each of these are 0.05 millimeters, right? And then what you're going to see are cells within here. And then you simply count the cells, right? Everything that's in. General rule usually is if it's touching or if it's kind of like half in, then you count it. If it's not touching, then it doesn't count. Okay, and you just simply count the number of cells that are within the square. It's easy, right? So the major advantage is it's quick. I mean, literally you take your sample, pop it in, right? Within minutes, you have your results, right? It's relatively inexpensive. All you need to do, okay, relatively, yeah? Because you're gonna need one of these, which costs hundreds of dollars, right? But other than that, it's relatively inexpensive. And then, of course, you get your numbers immediately. And, of course, you get actual cell number. And that's the most important thing. You see the cells. You know exactly how many cells there are. Disadvantage is that, when there are just a few cells, if they're just like 3 or 4 cells kicking around, because of that whole distribution and probability, right? 3 cells here versus 8 cells there, it's going to be really hard to tell that difference, right? So when the cell counts are really low, they're not terribly accurate, right? Just because of this probability. So what you'll have to do is count many, many, many squares to try to account for that. Other issues, dead cells and debris can look the same, right? So if 2 of these cells are dead, you're actually adding the dead cells as part of the count, right? So sometimes that's not useful either, right? So typically you'll do this for things that are in log phase because that's the most accurate. And then if these guys are moving, if these are motile cells, they're hard to count, right? And if you ever try to count, guppies in a guppy tank, anytime you have more than 10 guppies, it's really hard to count because they just don't stop moving, right? So that's always going to be an issue. And then for all you number phobes out there, you're going to have to do some math, right? Warning, here it comes. So let's do some math. All right, we're going to take this nice and slow, right? So we are going to count the number of cells in this little square here, right? Remember, anything that's inside the square is going to be counted. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right. Sometimes I like to double check that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, good. So we have 10 cells. Okay, and we're just going to leave that number there for now. All right. Because we're doing this, trying to find the concentration, we need to know what volume this number of cells are at, right? So this is where our math is going to come in, right? Now remember, let's look at this here. Each one of these squares is 0.05 millimeter. So we're going to look at this total big square like this, right? So again, over here, right? Each little square is 0.05, and we have 1, 2, 3, 4, 1, 2, 3, 4. Okay, so I'm going to draw this square out here just so we can have our numbers with us. All right, and then I'm going to put it in my calculator, 0.05 times 4. We have here a square that's 0.2 millimeters and 0.2 millimeters. All right, just write the units. Who cares if you don't know what a millimeter is, okay? We're just going to leave it like that. So we have a square, 0.2 millimeters by 0.2 millimeters. Okay, and then hopefully we know how to calculate the area of things, length times width, right? So we go 0.2 times 0.2 in our calculator. So we have an area of 0.04 millimeter squared. And again, you don't have, who cares if you don't know what a millimeter squared is, right? 0.04 millimeters squared. Okay, so that's the area of our square. Good so far? Let's hope so. Now, this chamber, it's not included in the data here, but they'll always tell you about the depth, right? So the depth of the chamber is 0.2 millimeters, right? So that means that the chamber is between the space between the actual cells and the cover slip, right? So if we look at it from the side, here's our cover slip. This here is 0.2 millimeters, right? And again, every single one that you see is going to be calibrated. I'm going to double check this number because it wasn't written down. Okay, so that's correct, 0.2, right? Which then means now if we know how to do volume, right? Length times width times height, all right? So this is the length, this is the length times width times height, right? So that now gives us 0.2 times, and we can now do it times our area. 0.2 times 0.04. Now that equals 0.008 millimeter cubed. That's a 3. Okay, so now we have a volume of 0.08 millimeter cubed. Okay, here comes the crazy part now. To turn this into milliliters, all right, we simply divide this by 1,000. Who cares if you don't know why? All right, I don't care you don't know why. This then now gives us 0. And we're going to add 3 zeros to this. 1, 2, 3, 4, 5, 0.08 milliliters. Okay, so our volume in this square here is 0.000008 milliliters. Okay, just count the zeros, right? We have 2 zeros here, and then we're going to add 3 zeros in front of that. Okay, remember we had 10 cells, right? So that means we have, I should have planned this space better, 10 cells in 0. I'm going to count 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Notice I don't say 0, 0, 0, 0, 0, right? Because that's confusing. I just count zeros. 1, 2, 3, 4, 5. Then I go 1, 2, 3, 4, 5, 8 milliliters. And that gives us our grand total of, let's put a line there. I should use my calculator for this just in case, right? 1, 2, 3, 4, 5, 8. 1,250,000 cells per mil, or 1.25 times 10 to the 6 cells per milliliter. That's terrible, but that's okay. You can see it. This is the exact calculation that's in your book. Right, so in figure 9.8, you can kind of follow along, right? So I know it's math. Okay. Again, at this stage, maybe if math kind of is a phobia to you, I'm not really expecting you to know why we're doing this. Just punch in the numbers in the calculator, right? Just be aware that you got to figure out how long or how big one of these squares is, right? Using the measurements that are there. The depth is always going to be slightly different, right? So make sure you find the depth. And then you simply go length times width times the depth. That'll give you the area. Divide that by 1,000. Who cares where this number came from? That'll give you the volume that all of these cells are in. And then you simply count your cells, divide it by this volume to get yourself this number, right? So this means that even though you only counted 10 cells, you actually have 1.25 million cells per milliliter. Tons of cells, right? And that's kind of what bacteria do. If you don't quite get this, rewind this, watch it again. If you don't quite get that, then post it on the discussion board, and then hopefully someone can help you figure it out. All right, sometimes when we are doing this, we just talked about this idea of, how dead cells can kind of affect this, right? You can actually make this a little bit better by doing what's called a viability stain, right? You can use color to differentiate live and dead cells, right? And this combination of colors will do one of two things, right? So we have two stains. The green stain will stain DNA, and it's a small hydrophobic stain, so it actually can cross membranes, right? So this little green stain will cross membranes, right? So it'll stain DNA, right? The red stains also stains DNA, but it cannot cross membranes, right? So remember, in a living cell, we're going to have cells that have intact membranes, right? Which means then that the red stains color only the dead cells, right? Because remember, they cannot cross membranes, whereas our green stains will only be able to, will color the live cells because they can cross membranes, right? So now when we're doing a count, we can actually, not only can we get a count of the total living cells, we can actually also see the number of cells that have died in this place, right? So if you're trying to do some sort of quantity over time, what's happening to the population, you can actually start to see how fast the dead cells are accumulating. All right, so that was the direct microscopic count. I hope your brain is still okay and we're okay to move forward. The plate count, sometimes called a viable plate count, is a really powerful technique because, as the name implies, a viable cell. This means that we're going to count only live cells, right? So it can be quite accurate when we're doing this because only live cells are counted, right? Basic premise here is that we take some microbes, we put it onto an agar plate, we let them grow, they form these colonies, right? Now, colonies are these mounds of like billions, bajillions of cells. But because, these billions of cells came from, presumably from a cell that is divided many times, kind of forming this mound on top of each other, right? Each colony has come from a single cell, right? So when you're counting colonies, we often just pretend that they are cells, right? Because of the fact that, if there are two cells that were kind of close to each other, they're going to form two colonies that are going to grow on top of each other. It's sometimes a slight under representation of the total number, right? And typically we don't count them as cells. We call these things colony forming units, a unit that has formed a colony, right? Plate counts are extremely accurate, right? However, so let's go to our advantages. They're extremely accurate because we count only our live cells. However, it does take time for this, the colonies to form, right? As we saw, it takes 20 to 30 generations for us to get some growth, right? So, that typically takes about at least 16 hours or so sometimes maybe 12 under extremely ideal conditions, and sometimes longer depending on the microbe that you're trying to count. So this is not the kind of data that you get today, right? You do the work today and then you get your data tomorrow. It's a lot of work involved, as we will see, right? And it only works with cells that grow in a lab. You have to be able to have conditions in which the cells will grow. If you have cells that don't grow in a lab environment, you can't use a plate count, right? So let's get back to this lot of work involved, right? Oftentimes cell numbers are really high, and when they're very high, we have to dilute them, right? And what does diluting mean? Yeah, we have to lower the count prior to plating because there's only so much space on an agar plate, right? So here's a figure, right? We have our bacteria growing in liquid media, and then what we end up doing is we take 1 ml of this and mix it with 9 ml of water. We mix that, we take 1 milliliter of this, mix that with 9 milliliters of water, 1 and 9, 1 and 9, 1 and 9, right? And they're trying to depict this here using color, right? So we have this kind of dark yellowish thing broth here, and then as we start to dilute, we're getting less and less because we're mixing it with water. We're cutting it, we're diluting it, making it less and less and less and less and less, right? And then each one of these is a 10-fold dilution, right? So we have 1 mil with 9, that's 1 in 9 plus 1, that's 10 total. So now we have 10 in here, so that's 1 in 10. So this is 10 times less. Yeah? And then we take 1 milliliter of this, mix that with 9 milliliters of water. Oh, did I give the math warning? Warning. Math warning. 1 milliliter with 9, that's once again now 1 in 9 plus 1 is 10. 1 in 10. This, 10 times less. Overall, it's 10 times 10 is 100 times less. Once again, 1 in 9, right? That's 1 plus 9 is 1 in 10, 10 times less, or 10 times 10 times 10, 1,000 times less than our original. This then gives us 10,000 times less than original, 100,000 times less than original. And typically when we do these dilutions, we do these in multiples of 10. Now we have to do this because when there are tons and tons of cells, we call this too numerous to count. This is bacteria that are growing all over the plate. It's just so swarmed that we cannot count them. And even when the colony counts are really high, something like this, 389, notice how they're all like jammed on top of each other. Really difficult to count, right? However, if we have too few colonies, like here there's 2, you know, the difference between 2 and 4 can be immense, right? But it's so easy to get that small difference whenever we're doing the work. So statistically, we like plate counts that have plates that lie somewhere between 30 and 300 colonies, right? And those give us the most accurate counts. So can you guys see what's coming up? Let's do one of these things, right? Let's try to figure out how much bacteria there is in this original broth culture, right? So remember, 30 to 300 colonies is what we're looking for. So what we're going to do is strike. That's 389. That's more than 300. That's 50. That's between 30 and 300. So that's probably the one we're going to use. 2 is less than 30. So strike. All right. So that's what we're using here. Okay. Now this 0.1 indicates that we took 0.1 milliliter of this tube and stuck it on the plate. And then we got 50 colonies, right? Remember, colonies, we can call that as essentially being cells or CFUs, right? So this means that in 0.1 milliliters, we had 50 CFUs, okay? So we have in this tube, where's my cursor? I lost my cursor. We have 50 CFU per 0.1 milliliter. Okay. Now, things per 0.1 milliliter are impractical, so you simply take your calculator, you take 50, divide that 0.1, and then what we then get is a concentration of 500 CFU per mil. Okay? So that number then represents that in this tube, we have 500 CFU per mil. Okay, but that's not the tube that we want, right? We ultimately want to get to here. You can cheat and jump. I don't like cheating and jumping, right? Because cheating and jumping doesn't give you 100% confidence, right? So what we're going to do is we're going to go one at a time. And remember what we did here, right? We said that this tube was 10 times less. This tube here was 10 times less than this one. This tube here, 10 times less than this one, and so on and so forth. So if we go backwards, that means this tube is 10 times more than this. This tube is 10 times more. This one is 10 times more, right? So all we're going to do moving forward here now is we're going to add zeros. So if that's 500 or that's 2 zeros, we're going to add a third zero. 1, 2, 3. Remember, I don't say 0, 0, 0. I just count zeros, right? So then this one here will have 4 zeros. 1, 2, 3, 4. This one here will have 5 zeros. 1, 2, 3, 4, 5. And then our final, this is the one we want to end at, has 1, 2, 3, 4, 5, 6. And then we go 3 and 3. So then that gives us a final answer of 5 million, or 5 times 10 to the 6 CFU per mil. Sorry, final answer. Again, if this doesn't fully make sense, just go back, right? Work your way through from the beginning, right? Go back and watch this again and just follow the steps that were outlined here, and then we should be able to get our way through this. If it still doesn't make sense, post it in the discussion board, right? But really the best way is don't just try it, and just like with the Petrov-Hauser chamber, don't just try it and say, that was too hard, I didn't get it, and then immediately start asking questions, right? The power is in trying to figure this out yourself. Rationalize what is happening. Don't memorize the steps, right? Rationalize it. What is happening every single time we do a dilution, right? We're going 10 times less. Do you understand why it's 10 times less, right? 10 times, 10 times, 10 times. Just try to rationalize that to yourself, right? How do we get these 50 colonies? Well, these 50 colonies came from this 0.1 mil, right? And remember, this volume can change, right? So just understand that it's 50 per the volume that we put, right? Because we took 0.1, that means in that 0.1 mil, we have 50 CFUs, right? And then once we get that number, then we just start adding zeros because we knew everything going down was 10 times less, while going back up, it's got to be 10 times more. One other last thing about the plate count is some samples with very few cells may not have enough microbes to count, right? We can realistically only plate 1 milliliter of a sample onto a plate, right? So, which means then that if there are very few cells, this is kind of a hard technique to use, right? But what we can do is we can concentrate samples using membrane filtration, right? So we talked about, I think was a couple modules ago, where we have membranes that can catch microbes or catch bacteria, right? The holes are just too small for bacteria to pass through. So we can actually take, 100 milliliters. We can take a liter. It really doesn't matter. As much volume as we want. We pass it through the membrane, the filter, and then the bacteria, they get stuck on the filter. Then we take that filter, put that onto an agar plate, and then we can count those cells. So we can actually take a large sample volume and put that in, right? And oftentimes for water samples, especially ones where you don't expect there to be a lot, like drinking water, for example, oftentimes you'll filter large volumes so then you can get a much more accurate count, right? Because you're not going to expect to have a lot of bacteria there. All right, finally, we have our most probable number, right? As the name implies, right? Already we can see one of the disadvantages, right, is that it's a probable number. It's an estimate, right? So we're not getting an actual, definitive. We're getting this estimation, right? But sometimes really that's all you need. Sometimes it's sufficient because for certain things, especially like food samples, it's like this range, there's going to be so many other things going on that, this range is sufficient. Water samples, oftentimes very common for most probable number. It's useful for when cell counts are really low, right? So, if you don't have a filtering contraption, right? So many of us don't have the ability to filter things. Most probable number is often sufficient, right? And the nice thing is there's this chart that's been produced that then allows you to then figure out what the probability is, right? So even though there's math involved, some math people have done the math for you, right? So it's actually pretty easy to do. The other disadvantage is that because we are dependent on growth, it does take time to see that growth, right? So it's not like we can see growth immediately. It does take a little bit of time, right? But basically what you do is you take this pond water, or in this case, this would be pond water, and we put 10 milliliters into this tube. We take 1 milliliter, put it into these 5 tubes. We take 0.1 milliliter, put it into these 5 tubes, let it grow for a day, right? And then we count positives. And in this case, yellow is positive, right? And so there's this, again, this probability, right? So remember these numbers: 10 milliliter, 5 positives; 1 milliliter, 2 positives; 0.1 milliliter, 0 positives, right? So let's consult our handy chart, right? And remember, 10 milliliter, we had 5 positives; 1 milliliter, we had 2 positives; 0.1 milliliter, we had 0 positives. So let's go down our list and then start looking. We got 5, 2, 0. Oh, look at that. 5, 2, 0. Good. Which then means that the most probable number is that there are 49 cells per milliliter, probably, right? And they even give you like this 95% confidence limit, right? So if like, well, it's 49, but it could be as low as 17. It could be as high as 130. And then within this range, you are 95% confident that your number lies somewhere within there, right? And again, for certain things like water samples where you're not really expecting to have a lot, and there's kind of this range, hey, you're allowed to have a certain amount of bacteria, and then you can swim in it. It's totally okay, right? Something like this is sufficient. And then if you start getting into these kind of high numbers, like right on the border of what you might think might be acceptable, at that point, then you would give that water sample a further look using different techniques, right? But sometimes an approximation is more than sufficient. All right, so those were our direct methods, right? We also have indirect methods, right? Using things called turbidity, dry weight, and cell or metabolic activity. Remember here, we're not counting cells. We're using something else to approximate or equate cell number. And the most common of these would be turbidity, right? And so the idea with turbidity is that we have this idea that the cells, even though they're small, they'll hit light and they'll scatter, right? So as you start to increase the amount of cells in a sample, the cells will start to scatter light. So we take this machine called a spectrophotometer, right? Kind of a funny name, or kind of a long name. Spectral referring to the spectrum of light that we can shine, photo referring to light. So let's say this together: spectrophotometer. One more time: spectrophotometer. Some people call it a spectrophotometer, right? And then so this machine will shine light. It has this light detector, right? So we're going to shine our light through here. And then if the sample is completely clear, there's no bacteria in it, we're going to collect a certain amount of light. The machine knows how much light it sent. It knows how much light came back. And it says this one has 0 bacteria. We call this absorbance or scatter, right? They're kind of opposite measures. Absorbance means that this sample had 0 absorbance or 0 scatter, right? Sorry, transmission is sometimes it's referred to as transmission. So 100% transmission. In this case, it would be 0 absorbance, right? Whereas over here, we have some bacteria present, right? As we shine our light through, some of this light scattered. So then this machine would say, some of the light got absorbed by the sample. Really got scattered, right? But some of it got absorbed. The advantage is it's really easy to perform. It doesn't really take much of a trained hand to be able to do this, right? You just stick it in this machine, close the lid, bang, read your number, done, all right? Disadvantage, it's most accurate when there's kind of a larger cell number, right? When cell numbers are super small, it's hard to see because in part, debris can scatter light. As cell numbers start to get really high, it becomes also less useful because dead cells will start to scatter light, right? And ultimately, it doesn't give you cell number. You can plot, you can do a cell count, and then do a turbidity reading, and then plot a graph, and then try to equate cell number with turbidity readings, right? And then use this graph to figure out, so you can take a turbidity reading and find out how much bacteria that equates to, how many cells there are, right? But it really only works for that one specific bacterium under that specific condition, right? So again, it doesn't give you a cell number, but sometimes that's all you need. If you're going to do a growth curve, if you're going to measure the amount of bacteria, and you're interested in to see whether the numbers go up or down, and you're going to do this every 10 minutes for 6 hours, turbidity is what I would do because it's so fast, so easy, right? And really all you want to do is relative, right? Are the numbers increasing? Are the numbers decreasing? Two other indirect methods are dry weight or metabolic activity, right? So dry weight is really literally what it sounds like. You're going to take your microbes, you're going to dry them, you're going to weigh them, right? It's not super duper useful except when you are growing filamentous microbes, right? Because filamentous microbes, they're hard to count. They're long filaments like we saw with the fungi, especially useful for fungi. It's really hard to tell where cells begin and end. So filamentous microbes, weighing is a great way to do this. However, the drying has to be performed accurately, right? If you leave even just a little bit of water left behind, that can confound your data, right? So you definitely don't want to do that. So drying must be performed accurately, right? But if you have this standard protocol for drying, and if you're growing fungi, dry weight would work great. Metabolic activity is this another way where there's this assumption that the more microbes are present, the more metabolic product they're going to consume or produce, right? So for example, if you have a room with 10 people, there's going to be some level of carbon dioxide produced, right? Because we're going to do our respiration, we're going to produce carbon dioxide. Well, if you have 100 people in that same room, you will presume that more people are going to produce more carbon dioxide, right? So if you had some sort of monitor to measure how much carbon dioxide was being produced at any one time, you can then get yourself this approximation for the number of people in a room, right? Or at least relative numbers, right? Similarly, you could also have food out there, right? So, if you put out 30 muffins and at the end you can see that, 15 are left. The next day you put out 30 muffins and then they're all gone, right? You can maybe presume that even without having been to the meeting, you can presume that, the second day we had more people present at the meeting because it ate more of the food, right? The major advantage of metabolic activity is when cells can't be cultured, right? Because remember, the plate counts and all those things require really that cells are alive and active, of course, but also that they can grow in the lab. Sometimes we can't grow them in the lab, right? But all cells can perform metabolism. So going back to that example of where they revived cells that were 1.2 million years old, right? They took that sediment sample and then they sprinkled various food and stuff on there, figure out a way to measure consumption and metabolism, right? And then so they didn't actually see the cells, right? But they saw evidence of metabolism, right? They saw evidence using this metabolic activity. And the other cool thing is you can actually do it in the environment. So you can take the sample with you back to your lab, right? Or if you have, you can also take your reading or your tool to the field and then perform the metabolic activity right there in the wild, right? Disadvantage here is that, you don't really know what you're working with, right? They're, mixtures of, oh my goodness, that should say data, not date, right? I'm going to change that. Can I? Yes, I can. Oh no, that was terrible. Data can make it less reliable, right? Because if you're going from one sample to another sample, the populations of bacteria might be different from that one sample to the other sample, right? So sometimes the differences that you see might just be because there are different microbes present, not because of the fact that the numbers are different, right? So, for all these methods that we just talked about, there's always going to be, pros and cons, hits and misses, right? So that's definitely something that you must keep in mind. Couple of things. I don't want to talk too much about biofilms, right? But they are very important, right? So biofilms are this complex ecosystem of microbes which allows many different microbes to live together, right? They can really form on any environment with some sort of moisture, including humans, right? And we all have biofilms growing on us, as we will see very soon. So biofilm formation, again, it's a lot more complex than it makes it seem here, right? We're not going to talk too much about it, right? But basically, biofilm formation always starts with, that one. There's some initial colonizer that just decides it's tired of running around, right? And it wants to stop and grow, right? So these initial colonizers, they say that they become sessile, All right, and so they attach to something, and they'll attach and they'll fall off, and they'll attach and they'll fall off. But at some point, they're just tired of that. They just want to attach and stay attached. At which point they become sessile, right? Then these cells start to secrete this extracellular polymeric substance, right? Extracellular meaning out of the cell, polymeric, bunch of polymers. And it's really different and varies according to whatever the microbe is, whatever the environment that you're in, right? So it's going to be some sort of EPS. As this stuff gets secreted, right, and of course it's going to be sticky, we have our secondary colonizers. So then these secondary colonizers will attach, and then they'll start sticking to this EPS, right? As the secondary colonizers are there, sometimes they'll attract other colonizers, and then next thing you know, you got this vibrant community of microbes all kicking around, all sticking together, all metabolically cooperative. And oftentimes here you'll see cases where waste product of one microbe becomes the food of another, right? And everyone is talking to each other. Everyone's doing these things. Eventually, some of them get tired of big city living, and they're going to move on and then form their own new biofilm or just go ahead and spread their wings. Now, one of the ways that this stuff is regulated is via this thing called quorum sensing. All right, a quorum, for those of you who are familiar with like meetings and stuff, means that there's like a minimum number of required people present. In this case, it would be microbes, right? So quorum sensing is this mechanism for microbes to be able to detect the amount of other microbes in the environment, right? The way they do this is they have this thing called an autoinducer. Basically, usually it's a short peptide or some sort of molecule that they just kind of spit out, right? So microbe will be present, it'll just spit out this autoinducer. Sometimes it's meant to be within one species, so only like the same species can sense the autoinducer, right? It's like an inside joke. All these other ones, they see these short peptides, they don't know what to do with it, right? But all the ones that there's one species are like, we're speaking to only ourselves, right? And sometimes it's designed to talk to many species, cross-species communication, right? And then when the autoinducer builds up to a certain level, right? Because once again, the more of a species are present, the more autoinducer is going to be produced, right? At some point, they're going to reach quorum, and then stuff will happen, right? So oftentimes here, pathogens, they'll use things like quorum sensing to hide. They're non-pathogenic until they get to a certain number, and then they break out, and then they attack the human host, right? And then they do their thing, right? And in fact, some therapeutics are designed towards binding up that autoinducer. So even though there are enough bacteria present, right, the autoinducers are not being released, or at least being recognized, right? And then so the pathogens, they don't become active and they don't cause disease. There are biofilms found in humans. Some are beneficial, some are harmful, right? So our normal microbes in our mucosa lining, right, that they form this biofilm, which helps them prevent attachment. And then this biofilm is good in excluding bad microbes, right? But there are bad biofilms as well. So plaque, that's the one that we all have, right? That's a biofilm that allows microbes to stick to teeth. It's started by some Streptococcus species in your mouth. They metabolize sucrose, right, the sweet sugar, and then produce that biofilm that allows it to stick. And then in a medical setting, bacteria can form biofilms on medical devices, especially stuff like catheters, artificial joints, even contact lenses. And then these things lead to chronic infections because the biofilms will protect the pathogen from things like antibiotics, really hard to clear out. Okay, that's enough for now. Actually, I got to run off to a meeting, so I will definitely take a break. And then you guys can take a break, and then we'll see you guys very soon. All right, I'm back. Hopefully you guys didn't miss me too much. My meeting was extremely exciting, but here I am. Now we're going to do some even more exciting stuff and continue to talk about microbial growth. Continuing on with the requirements for oxygen, right? So actually the next little while, what we're going to talk about are just what are the requirements for growth? How do we determine, how do we measure them? How are microbes affected by them? And we're going to start off with oxygen, right? So aerobic metabolism, as we recall, provides more energy than anaerobic metabolism, right? Because more ATP is produced. And of course, in the game of things, in the game of life, more ATP equals much more robust growth, right? However, the thing with oxygen is that it's actually quite toxic. The aerobic metabolism process produces all these things that we call reactive oxygen species, these molecules of oxygen that end up reacting with things and producing a lot of cell damage, right? So in order to be able to survive in an oxygen environment, you must be able to detoxify these toxic oxygen species. And if you cannot detoxify these oxygen species, then you cannot live in an environment that has oxygen in it, right? So you must grow anaerobically. So oftentimes, actually, when we talk about anaerobic growth, many times when they're growing anaerobically, it's because they have to. It's because oxygen is so poisonous to them that they can't survive, right? And some very common environments that are anaerobic would be these swamps. I think we talked about these swamps that get all anaerobic, end up smelling a lot like rotten eggs and sulfides and things like that. And then the rumens of cows, even our intestines, right? We have a lot of bacteria that grow in our intestines, which is an anaerobic environment. And they're there in part because of the fact that, they have to be. So we can determine whether or what the requirements for microbes' oxygen levels are using something called a Thioglycolate tube. And we don't want to get too much into what thioglycolate is exactly, right? But basically, it's a technique for allowing us to see the oxygen requirements of a microbe. Within this tube, there's an oxygen gradient. So at the top of this tube, there will be high levels of oxygen as it diffuses down. And at the bottom of the tube, there will be no oxygen as the oxygen doesn't quite get to the bottom. And then so based on where microbes are able to grow throughout the tube, we can categorize microbes into 5 groups: obligate aerobes, obligate anaerobes, facultative anaerobes, aerotolerant anaerobes, and microaerophiles. So let's define these 5 groups, right? The word obligate means that you must, right? So obligate aerobes must grow with oxygen. Obligate anaerobes must grow anaerobically. Facultative anaerobes, now you can almost think of this like facilitative. It's not the same thing, right? But facultative means you can do both. Aerotolerant anaerobes grow anaerobically, but they can tolerate oxygen. And then microaerophiles, they like small amounts of oxygen. Okay, so based on that, let's look at thioglycolate medium. Right, so here these little red dots indicate bacterial growth. And this is our thioglycolate medium. Right, high amounts of oxygen, low or no oxygen down here. So based on the fact that these microbes are growing only at the top, which of these categories are we looking at? If you said obligate aerobes, that's correct, because these are growing in an area and only in the area where there's a high amounts of oxygen, right? These are our obligate aerobes. They have to grow where high oxygen is because they must grow with oxygen. And consequently, they also have enzymes that detoxify oxygen, right? So they can survive in this oxygen environment. All right, B, we have microbes that are growing only at the bottom. So which one do you think these are? If you said obligate anaerobes, you're correct, right? So these ones, they have to grow anaerobically, right? So they grow right at the bottom. And thus, and the reason for that is because they lack enzymes to detoxify oxygen, right? So they are unable to survive in an oxygen environment, which means actually oxygen kills them. It's not like they can't grow with oxygen. It means oxygen kills them, right? Generally, they're going to grow a little bit slower than aerobes because of the fact that they're doing anaerobic respiration, which we learned produces a little bit less oxygen. A little bit less ATP. But, they still do respiration. This one here is interesting. We see growth throughout the tube, but notice how there's this cluster of growth at the top and then a little bit less growth, but still found throughout. These ones would be facultative anaerobes because they can grow both with and without oxygen. However, when they're growing without oxygen, they're typically using fermentation or respiration, which means, of course, that they produce less ATP. Thus, they will have a little bit less growth when growing without oxygen, as opposed to when they're growing with oxygen, they're able to grow much better, right? So they grow faster with oxygen. They prefer that, right? But in the event that oxygen is not available, they can still grow. They just don't do it quite as effectively. This one looks kind of similar, right? Notice how we have growth throughout again, right? But in this case, there's equal growth throughout the whole tube. So these would be aerotolerant anaerobes, right? So these are typically our obligate fermenters. And remember, fermentation doesn't use oxygen, doesn't care about oxygen, right? So in this case, with oxygen, without oxygen, growth is going to be exactly the same because they don't care, right? Of course, to live in this environment, they do have to have enzymes that detoxifies oxygen, because the oxygen doesn't kill them, but they're more or less irrespective. They are aerotolerant. And look at this one here. There's only one left, so obviously we can figure out which this one is, right? But they're not going quite at the top, a little bit less down. So these are our microaerophiles. And the microaerophiles, they require oxygen. They do aerobic respiration, but they cannot detoxify high concentrations of oxygen. So too much oxygen is bad for them. Good. I do want to show you real quickly when we grow these things in the lab, right? Aerobes are easy to grow. Facultative anaerobes, they're easy to grow. Aerotolerant anaerobes, easy to grow. It's those obligate anaerobes that are really difficult because passage in this oxygen environment ends up killing them, right? So you have to have special things with them. So over here we have an anaerobic jar, right? This packet right here is able to remove oxygen and produce this environment, typically of hydrogen and carbon dioxide, that allows for anaerobic growth, right? So then these agar plates are stored in such a way that anaerobic growth is supported, right? So then typically that's how we do it in a lab environment. And for the extreme obligate anaerobes, ones that absolutely cannot even be passed for a second in the air, we often work with this anaerobic chamber, right? So this, even though it's facing out, this is actually where your hands go in. And at the end of these are gloves. You stick your hands in there and then you manipulate in this environment that's 100% anaerobic. Okay, so based on this then, we can define a few things based on oxygen requirements. We can define them as being the optimum oxygen concentration, right? Which, it's pretty straightforward as the name implies. It's the ideal concentration of oxygen. Minimum permissive oxygen concentration is the lowest concentration that will allow growth, and the maximum permissive oxygen concentration is the highest concentration that will allow growth, right? Because when we say things like, oh, we are in a 20% oxygen environment, doesn't mean that 20% is the only thing that we need. We can probably work a little bit higher. We can probably work at a little bit lower concentration, right? And then that will define our oxygen requirements. Okay, so let's talk a little bit about the reactive oxygen species. So these are the most common species that are there, or that we will talk about, right? Singlet oxygen, superoxide, peroxides, hydroxyl radical, hypochlorite ions. These weird dots, by the way, are something, right? So these are like missing electrons. That's kind of what that means, right? And all of these are very unstable. Think of these things as molecules that just hate the world. They're just angry at the world. They're totally unstable. And the only way that they can find fulfillment is by just wreaking havoc on everyone else, by taking away other people's electrons, right? So that's what they end up doing, right? So they end up just reacting with your DNA, your membranes, your proteins, and just causing this damage, causing everything to fall apart, right? Ultimately killing the cells. So in order for us and any organism that can survive in an aerobic environment, we have to be able to detoxify these things. And so there are three main enzymes that exist. You don't have to know the full mechanisms of these. I will point out some of the things that you do need to know. So we'll start off with superoxide dismutase. It's primarily used for the superoxide anion, right? It's very important. This is a very important enzyme for survival on oxygen. In fact, almost every organism that survives in an oxygen environment has some form of a superoxide dismutase, right? The reaction here you can see as pointed out, but this is the part that I want you to know. One of the things that it does produce is H2O2, which is hydrogen peroxide, right? And hydrogen peroxide itself is a reactive oxygen species, right? And there are two mechanisms to convert or to detoxify the peroxide anion or hydrogen peroxide, one being catalase and the other being peroxidase, right? So both of these will take hydrogen peroxide and then detoxify it. Main difference here being that catalase produces oxygen, whereas peroxidase does not, right? So we actually have these enzymes, one of these two enzymes, catalase and peroxidase. right? Which one do we have? Do you know? Here's how you can tell, right? So if you've ever used hydrogen peroxide as a disinfectant, right? So you have a cut, a little bit of blood's coming out, and then you put some hydrogen peroxide on there. What do you guys notice, or what do you guys remember happening? Little bubbles, right? And people used to say, oh, the bubbles means that the peroxide is working and killing the microbes. It's actually not true. The bubbles are because our red blood cells have the catalase enzyme, right? So the peroxide then gets broken down by the catalase in our red blood cells to produce water and oxygen. And this oxygen being formed, it's literally this reaction happening right in front of you. If you sniff it, right, you are breathing very freshly produced oxygen, never before breathed by anything else, right? So we are catalase containing organisms. Obligate anaerobes, of course, have none of these enzymes because they can't survive, and they must be in an environment that does not have oxygen. And then there's a special group called capnophiles. All right, they are anaerobic, but they also require high concentrations of carbon dioxide, and this carbon dioxide is very important for them. Okay, so that's the requirements for oxygen. Going through our list, we're going to continue and talk about the effects of pH. Right, and so pH, we're going to get into like just a teeny tiny bit of the chemistry, right? So pH is a function of protons, right, or hydrogen ions. We've seen this before, right? And our pH scale is a scale that goes 1 to 14. We don't have to talk about what the numbers mean exactly, because they do mean something. We're going to keep it nice and simple. The middle, pH 7, is neutral, right? So 7 will be a neutral pH. And then we go less than 7 is acidic, greater than 7 is basic or alkaline. And of course, the more less than 7, the greater we are to 1, the more acidic. The greater we are to 14, the more basic or alkaline. And pH is important because extremes of pH affect things like hydrogen bonds, lipids, because the high pH tends to hydrolyze lipids or break them apart. Proton mode of force, right? If we are in an environment where there's a lot of protons already on the outside or very few on the outside, it can really affect the proton mode of force and thus ATP production, and then proteins, right? We talked about how proteins require a very specific set of functions or a specific environment to work in. So if you change that pH significantly, proteins will become denatured, right? So then based on an organism's ability to grow in a certain pH range, we can once again define certain terms. And this is going to sound like what we just talked about, right? But we have our optimum growth pH, a minimum growth pH, and our maximum growth pH, right? And so this is, pretty straightforward. I don't think we need to spend too much time talking about it, right? But this pH ranges then has an effect on the way we classify our organisms. Right, so we have what's called most microbes, are the neutrophiles, right? So most of them have an optimum growth pH of pH 7. Most things like neutral pHs, right? And then we can go a couple of pH points plus or minus to define our maximum and minimum pH environment, right? Some microbes, they love the acidic environment, right? So acidophiles can grow at low pHs, right? Now, of course, because of this effect then on our proton mode of force, right? One way to survive is to have membrane pumps that help maintain this proton mode of force, right? So it can really goof around with the proton concentrations. And then phospholipids are modified to help maintain fluidity, right? Because we talked about how changes in pHs will affect lipids. And it's the same for alkalophiles, right? Those that grow at high pH, in order for them to survive, oftentimes they're going to have to maintain a proton mode of force, right? Because they're going to be in the opposite situation as acidophiles. And they're going to have to be able to modify their lipids and proteins, right? So their protein structures, their lipid structures are designed so then they can maintain structure at high pH. I do want to talk a little bit about acidophiles. Acidophiles are wonderful microorganisms because many fermenters are acidophiles, right? And we talked a little bit about fermentation, right? What is, it's with respect to competition to aerobic respirers, right? So what's the major competitive disadvantage that fermenters have compared to those that do aerobic respiration? Anyone? Who's got it? Right, they produce such little ATP, right? Only 2 ATP compared to 38 ATP for our aerobic respirers, right? However, don't forget now, our fermenters have a certain advantage in that their products of fermentation are typically some sort of organic acid, right? We have lactic acid fermentation, butyric acid fermentation, propionic acid fermentation. And so over the course of time, as these fermenters are growing, they're going to continue to spit out these acids, and our pH is going to go from a nice neutrophile friendly 7 down to, and it can go down to as far as pH of 2, right? Which is important for us, right? Why would it be important for us? Well, lovely fermented foods, things like yogurt, pickles, sauerkraut, kimchi, if you're into that kind of stuff, right? So all these things are the process of fermentation. And the reason that they work, the reason that we're able to preserve the foods is because of this acidity that's produced by our fermenters, right? And then it keeps the foods from spoiling, right? And so that's one of the reasons why fermentation was developed or really invented over the years. Because fermented foods just lasted much longer, right? And of course, this is time before things like refrigeration and stuff like that, right? The Koreans, they fermented kimchi so then they can have vegetables over the winter when there were no vegetables available, right? And it lasted much longer. Fermented cabbage lasted much longer than raw cabbage, right? So, and then of course the acidophiles that produce these fermented foods, they're able to survive, and then they can outcompete those aerobic respirers who are typically responsible for the food spoilage. All right, our next characteristic that we're going to look at is temperature, right? Again, look at these definitions. They're going to sound very similar, right? We have our microbes that grow within a very specific temperature range, right? Things like low and high temperatures affect protein structure, DNA structure. All of these things are very important. And then so we can then once again define microbes based on the subset of growing conditions or growing temperature, based on their optimum growth temperature, minimum growth temperature, maximum growth temperature, right? And just like before, then we can define microbes based on these characteristics. Most are what we call mesophiles, right? Mesophiles like this moderate range. Oh, by the way, let's just define a few things on their temperature scale here, right? So this is in Celsius. I know it might be foreign for some, right? But let's define a few things. 37 here is, I'm just going to put BT for body temperature, right? Somewhere like around here, 20-ish is a room temperature. Our refrigerator typically tends to hover around like 4 Celsius. And our freezer typically tends to be somewhere on the -10 to -20. So that's our freezer. Water boils at 100. Oops, I don't know why I did that. So this is boiling. Hot water is somewhere around here, 50 to 60, something like that. Okay, then. Based on our scale now, right, let's define the mesophiles. They're probably the most significant to us because most human pathogens are mesophiles. And why would that be? Oh, because they grow ideally at body temperature. They love body temperature. They just want to be inside and hang out in this really warm environment that we produce for them, right? But they also grow very well at room temperature, right? So food spoilage organisms are also typically mesophiles. And so in general, mesophiles really have a big impact on humans. Psychrotrophs, kind of this moderate, right? It's not on our scale here, but psychrotrophs would kind of fall, in this temperature range here, something sort of like that. Maybe it's a little broad. Oh, there it is, 4 to 25, right? There are primarily food spoilage organisms because look where they can grow. They can grow very well at room temperature. They love growing there, right? But they can also grow in kind of cold environments, right? Kind of the fridge temperature. So even though the fridge, they'll be growing super slow in the fridge environment, they still can grow in the refrigerator, right? So, the psychrotrophs are primarily food spoilage organisms. Typically the outdoor microbes that are hanging out there, they tend to be psychrotrophs. rarely pathogenic because of the fact that they don't grow very well at body temperature, right? But some of them can produce compounds and toxins that, if left in the food, can make us sick, right? So maybe the presence of the microbe isn't what's going to make us sick, but they can leave behind things that will. On the opposite end, thermophiles, they must be in hot environments, right? So thermophiles and hyperthermophiles, they love super duper hot environments, right? Now, growth at extremes of these are going to require certain adaptations that allow them to survive, right? So when things are cold, things are slow moving, right? So lipids tend to be unsaturated, because remember, unsaturated lipids tend to have more kinks, keeps membranes more fluid. They'll actually produce these things called antifreeze proteins that prevent their cytoplasm from freezing by lowering the freezing temperature. Proteins tend to be more hydrophobic. Again, keeps it a bit more fluid when things are cold. And then at hot temperatures, things are going to be the opposite, of course. Lipids, a lot more saturated, keeps it a lot more fluid. DNA is GC rich. Hey, what does that even mean, right? GC. So this refers to our cytosine and guanine nucleotides, right? So DNA is very rich in G's and C's. Why would that help? Anyone remember? Yes, if you said that GC forms 3 hydrogen bonds as opposed to our ATs, which form 2 hydrogen bonds, you would have been correct, right? So remember, more hydrogen bonds means that the DNA is less likely to denature, right? So they tend to be rich in GCs, which keeps them from denaturing at high temperatures, right? And then proteins will have extra bonds in there, again, to help keep them from denaturing. And then, completing the circle on those 1.2 million year old microbes, these are hydrothermal vents that are found deep in the ocean. This is an environment that, even though it's deep in the ocean, it's extremely hot because it's this hydrothermal vent. And so all this life that you see here is sustained by microbes. Microbes are able to grow here, and then they produce organics that then all these weird little tube organisms and worms and stuff like that are able to feed off of. Osmotic pressure. So we talked about osmotic pressure before, right? Remember this difference in solute concentration then that allows for osmotic pressure, or greatly affects the water flow, right? And then recall that hypotonic, right, very low solute environments, they don't really affect bacteria at all too much, right? Because the cell wall helps protect from lysis, right? Because remember, in a hypotonic environment, that's low solute outside. That means high water. Water flows into the cell, right? Cells generally are protected from lysis because of the cell wall, right? But hypertonic is bad because hypertonic environments results. So that's, remember, maybe I'll draw that, right? So we have hypertonic, we have high solutes. I'm going to write it like that, which means low water outside the cell, inside the cell relatively, then we have low solutes, and then high water, right? High to low, water will flow out, and that causes plasmolysis, or shrinking of the cytoplasm, right? So it actually, the way we preserve foods using high salts or brines, right? That helps preserve the foods because of the fact that bacteria, they don't really have a defense against this, right? So bacteria are unable to resist this hypertonic environment, right? And thus they're able to, we can preserve foods from growth. Interestingly, I'm going to save that interestingly for the next slide because I feel like it's in the next slide. Halophiles require high solute environments for growth, right? So these are just like thermophiles, just like acidophiles, right? They don't like high salt environments, they need it, right? And because of the fact that they have modifications or adaptations that allow them to survive in these environments, right? So they typically tend to balance the outside environment by having or making high solutes on the inside. They have proteins that are modified such that they can work in these hypertonic environments, right? So if you actually want to think about it this way, if you have a halophile that has a whole bunch of solutes inside the cell, a hypertonic environment is actually isotonic, because remember, it's all a relative thing, right? So halophiles are interesting. They grow in these high salt environments, usually not super prominent for us. For us, more the halotolerants are going to be affecting us, right? Because halotolerant microbes can survive high solute environments, but they don't require it, right? So, our food spoilage organisms, they can grow in salt environments, right? But they don't need to, so they can actually do both, right? Bacteria on our skin, many of them are halotolerant because our skin is this salty environment, right? But again, they don't need it. Fungi, interestingly, here's our interesting fact. Now, fungi are halotolerant. They're also psychrotrophs, right? So, you think about, you leave some Chinese food in the back of your fridge, right? You leave some Jello. Next time you make some Jello, put a little bit to the side and just leave it in the back of your fridge. Give it time, right? And then what's the first thing that's going to grow on it? It's going to be some sort of fungus, right? And the reason is because they're psychrotrophs. They're able to survive or grow super slowly, but still grow in this cold environment, right? But something like Jello, it's high sugars, right? Which of course means that it's high osmotic pressure, right? Bacteria, they don't generally like Jello because of the fact there's just way too much sugar, right? It's this high osmotic pressure, right? But fungi, they can tolerate that, right? So, fungi really find this niche environments that they can grow in and then do some wonderful things for us, right? Like spoil our Jello. Related to osmotic pressure is both water and atmospheric pressure, right? So it's somewhat related, right? Moisture, we measure as something called water activity. So liquid water, which has nothing in it, has a water activity of 1, right? So bacteria, remember, they like a little bit of salt, right? Most bacteria prefer a water activity of 0.97 to 0.99, right? Kind of close to 1. And so one of the reasons that drying things preserves the food is because we lower the water activity by removing it, right? And so when we remove the water, we can prevent the growth of bacteria. And fungi, just those buggers, right? They can tolerate a much lower water activity. So they can tolerate as low as 0.75, right? So when you dry things like jerky, you got to make sure that you get it dry, right? So if you're a home jerky maker, dry enough is probably not good enough to keep it from fungi, right? So when you are doing home jerky, you want to make sure that it's dry, so then that nothing will grow on it. Barophiles are kind of interesting, more from an ecological standpoint. I don't want to talk too much about it. They require high atmospheric pressure, right? So, when you swim to the bottom of the pool and your ears hurt, right? Like way, way deeper than that, right? So they live at the bottom of the oceans. And I think that's all I want to say about that. So when we combine everything together, right? So now we know that there are certain factors of microbes that they require to grow. There are certain things that they can do. All right, we can put it all together, and then we can then design media or food sources for microbes in the lab that will allow certain types of microbes to grow, certain types to flourish, certain types to not grow at all, right? Generally all-purpose media can support growth of many types of microbes, right? So we have many different types of all-purpose media, but we can define these things into these two general groups called complex media and chemically defined media. Now in a complex medium, you have this undefined mixture of nutrients, right? Doesn't mean that it's bad, by the way, right? In fact, complex media tends to be one of the most richest food sources. But it's undefined because typically we use things like extracts of yeasts, meat, plants, things like that, right? So some common things are like soy. You just take soybeans, you dry it up, you grind it into a powder, soy extract, right? You take beef, you dry it, grind it into a powder, beef extract, right? Sometimes you have digests or yeast digests, right? Soy digests, right? Where you take these complex mixtures of proteins and you just cut them up into small little pieces, right? So when you're looking at the recipe for a medium, look for things like something extract, something digest, right? Things like tryptone, peptone, those are like weird protein mixtures, and those things are indicators of some rich nutrient source, but we don't really know exactly what's in it, as opposed to a chemically defined medium where we know the exact chemical composition, right? So this would be not just the recipes, but exactly how much. So if you need to say, for example, grow microbes that require a certain amount of phosphorus or a certain amount of potassium, well, you can calculate and you know exactly how much is in there. So look for things like chemical symbols, KH2PO4, right? And then a weight. So then you know exactly what's in there. Okay? So those would be all-purpose media. And then specialized media then exists for specific purposes. And we're going to talk about 3 types of specialized media. A selective medium is one that supports the growth of what we call our quote-unquote organism of interest, right? So basically, we have a certain organism. So for example, let's say I only want to look at gram-positive bacteria, right? So we can make a medium then that will kill gram-negative bacteria, right? Or inhibit them by maybe digesting their LPS, right? Or maybe by digesting outer membranes. It's all of a sudden now gram-negative bacteria cannot grow, right? Which then of course means that only gram-positives are going to grow. So we're selecting for some microbe that we want to grow. Kind of related to that is an enrichment medium, right? So an enrichment medium provides nutrients that help our organism of interest grow. Now, others are going to be able to grow as well, right? But sometimes the things that we want are going to be just in small numbers. So something like, let's say, a hydrocarbon eating microbe, something that eats crude oil. You're not going to expect that to be prevalent or very abundant in the soil environment, right? But what if you take the soil and then you feed it only hydrocarbons, right? Well, then just the ones that can grow in the hydrocarbon are going to be able to grow. They're going to multiply, right? Whereas the other microbes, they're going to have a much harder time. Maybe they can do a little bit. Maybe they can find a little bit of scraps here and there to grow on. But in general, they're not going to be able to do it as well, right? So we are now enriching this environment for our organism of interest, right? Similar, we could do this for things like autotrophs or for certain types of anaerobes or aerobes or whatnot, right? Differential media will allow things to grow, but our organism of interest will look different. And it's usually based on color. So, one of the classic lab examples is this medium right here, McConkey medium. And so what McConkey medium does, it's actually selective and differential. It selects for gram-negative bacteria, right? So the fact that we have 2 different types of bacteria here, the fact that we see growth indicates that both of these are gram-negative bacteria. And then the bacteria that we placed here and here are not growing. So these 2 bacteria are gram-positive bacteria, right? Because again, we're selecting for gram-negatives. McConkey medium also allows us to differentiate for this ability to ferment lactose, right? And it does so by turning the medium pink. So out of these 2, which one can ferment lactose? Well, this one right here, right? So this organism can ferment lactose, this one cannot, right? So we actually know then that this organism is gram-negative, cannot ferment lactose, whereas this organism is gram-negative and can ferment lactose. Right? And many different types of media exist that are selective and differential for various abilities. Things you want to look for are we selecting or are we differentiating bacteria based on growth? If things can grow and cannot grow, that will be a selective medium. And if things look different, that will be a differential medium. Okay, that's all we have for Module 5, right? Kind of outlining the requirements for growth. Now that we know that, what can we do to prevent microbes from growing, right? So what can we do to kill off microbes? How do chemicals work? What do they do exactly to attack microbes? Okay, and that'll be the subject of Module 6. So until then, I guess we'll see you in Module 6.